In this video, I will be walking you through the steps on doing a quantitative Western blot. And so first off, here in the project, we'll go ahead and click on Create New Experiment, Quantitative Western Blot. Now the steps involved with doing the housekeeping protein, the post-translational modification, and the separate target scan, all three of them are essentially the same process, the only difference being that how you import the images. But once you have the images into the analysis, the method for doing that set analysis is pretty much the same. So I'm going to use the post-translational modification, and since we've already imported these images into the image gallery, I'm going to click on that, going to scroll down to the image that I need, and click Select. And so my internal loading control in this case happens to be the total ERK protein. And then I'm going to add the 800 channel for my two versions of the phospho-ERK. And I don't have another image on here. If we did have one for the separate target scan, you can add that as well. And then go ahead and click on Start. And so this is going to be ERK, phospho-ERK. And you can write any kind of description from the protocol or anything you want in that area right there. And then go ahead and click on Next. And so then my internal loading control is going to be total ERK. And then my protein is going to be, actually I have two proteins that I'm going to quantify here. So this is going to be phospho-ERK 1. And then I have a second protein that is going to be phospho-ERK 2. And if you want to add any comments in here, you can also have that information as well. So for the boundaries on here, I do need to include the molecular weight marker because I do want to size these bands as well. So you click and drag just to the outside of your lanes. And if you have auto band finding set, it will try to find the lanes automatically for you. Now, if you find that it didn't find the bands correctly, you can go ahead and switch to manual, set the number of lanes, and then move or adjust them as needed. But since this did a pretty good job, I'm going to go ahead and stick with auto, and then click Next. And then if you have replicates on here, you can group these together to create these links, and that will be useful later on for the calculations that are generated. And then click Next again. So here we can enter in the sample information. We can give names to the lanes if we want to. We can add any kind of treatment information. We can also add the amount, concentration, or dose that was given during the experiment. If none of those four options fit your needs, you can click on this new value label. These are done in percentages by default, but you can also add micrograms, picograms, nanograms, and microliters. Now again, if none of those fit your needs, you can go ahead and click on New Unit Label. And so I'm just going to put in some values here. I'm going to say 100% treatment with my drug, and then you can either fill those in as you go down through here, or you can add in a dilution factor, or you can have it fill if they're all the same right here. But I'm going to go with a twofold dilution factor. Click OK. And then those values are filled in automatically. My 10th lane down here is my molecular weight marker, so I'm going to designate that as such right there. Go ahead and click on Next. And then for the molecular weight marker, I'm going to choose the Chameleon Duo 700 and then add my marker bands. So I click on those in those areas right there, and then click Done. And then I'm going to switch over to my 800 channel and choose the 800 Chameleon Duo and add my bands here. Now, something very useful here is that I hadn't extended my boundary high enough for that to be included, so now I need to edit this slightly. So if I click on Information, I can turn off that highest molecular weight, the 260, and then I'm going to give this a separate name so that I can use this later on if the same thing happens again. And now you see that the correct values are being displayed here on my molecular weight markers. So once I'm done with all that, go ahead and click on Next. And now we're going to quantify. So in the 800 channel, I'm sorry, the 700 channel, I'm going to click Find Bands. And it automatically places the band markers on there. And if you find that some of the bands need to be adjusted, either in the location or the size, you can do that on an individual basis right there. And go ahead and click Next. And this is just a review of the details. There's nothing really to enter or change at this point in time. And then so for my ERC1, I'm going to click Find Bands and position that right there. And it looks like it mostly found everything okay. I'm going to expand that just a little bit to include all of the band like that. And then I click on the P2. I click Find Bands and place those on there as such. And if you want to review any of this, you can click on the analysis table and look at the values for either one of these. They are average, so that's why you see that right here. We also get the standard deviations, and all of that information is included right there. Click Next. We have the charts, and so we're going to say that my first group is the control, and I do want to include my other replicates on here. And you can either look at it as fold change or percent change. You can either have it be a bar chart or a scatter chart. You can change the names of this if you would like. You can change the X and Y axis labels. But all of that is available right here on the chart screen. And again, if you look at the analysis table, you can pull up that same information as well. So then we're going to look at the review and the report. You see that it has the all of the information up here at the top. You can expand these if need be. It has the each one of the images. It has all of the data on here. Now, if you want to export these data out, you can click on this little arrow button right here, and it will export that as an Excel spreadsheet, and you can just save it wherever you would like. And same with the images. If you click on one of the images right here, you can export that image as a TIFF or in PNG. PNG also gives the option of 300 dpi, and so that's how you export the images. And as you scroll down here, you see that we have all of the data for the different proteins. It has a chart that we created earlier, and then it has a composite image at the end. You can add any kind of experimental observations that you would like, any next steps that need to be taken, and then a summary of the experiment. You can export this as a PDF report. The report generates just a PDF file that contains all of the images, all of the data tables, all the experimental conditions, all of that, and just puts it into a PDF file. You can export it as an experiment file, which allows you to move it to another computer, and if you need to do any further analysis, it's available at that point. Thank you.